Good morning, student. Today we are going to start new unit, that is sixth one, that is Callus culture technique. So in last lecture we have studied what is meant by callus and how it is formed. The callus is unorganized mass of cell that is produced from callus upon the addition or exogenous addition of plant hormone, that is addition of auxin and cytokinin. And the callus tissue is the result of influence of exogenous addition of hormone and the endogenous concentration of plant hormone, and that will result in cell expansion and cell division. So first we will see the morphology and internal structure of callus. You will get a question in exam, the morphology and internal structure of callus. So callus has no shape, it's irregular in shape, and the callus, which is derived from different plant, looks alike, but that can be distinguished on the basis of their morphological characteristic like texture, coloration, then hormone requirement, amount of water content, that is whether it is dry or wet. So we will discuss this morphology one by one. So first is callus texture. There are two types of callus texture: soft callus, that is also called as a friable callus, and another is hard callus. Hard callus, that is also known as a compact callus. So soft callus, which is friable in nature and made up of different mass of cells, that is heterogeneous mass of cells. And hard callus or compact callus, that consists of giant cell, tracheid cells, and those are closely packed cells. And the friable cells, friable callus, it is also used for suspension culture. So you can see in this image, so this is friable callus or soft callus, and this is compact callus or hard callus. You can see the cell has irregular shape, while those are not closely packed, and there are space present between these two cells. You can see in this image. While in compact callus, you can see there are some cells which has a larger shape, and those are closely packed. So you cannot see any large intracellular spaces between these cells. So the larger cells, which is termed as a giant cell, then you can see there is formation of tracheid cells. So this is the texture of callus tissues. Keep in mind the friable callus or soft callus, that is used for suspension culture. Then next is about coloration. So generally you will find the creamish color or white color callus. Sometimes there is development of green color callus, and that is due to the formation of chloroplastid in the cells of the callus tissue. Sometimes you will find the yellow color, that is due to the presence of pigments, and the that is carotenoid pigment. And sometimes what happens, there is formation of brown color pigment. So that is not due to any plastid or due to any pigments, that is due to the oxidation of phenolic compounds. And this oxidized phenolic compound, that is quinone, that will give brown color to the callus. So next is internal structure of callus. How you can observe this internal structure? That is with the help of light microscope. You can use compound microscope. You can use electron transmission microscope. So in that you can observe the heterogeneous cells, which is range from small to larger cell, having the dense cytoplasm, and some cells having vacuolated cytoplasm, and the shape is vary from spherical to elongate. So you can see in this diagram, so some cell has a oval shape and some are elongated. These morphological features, that is texture, coloration, Then hormone requirement, amount of water content, that is going to vary with the plant species, that is going to vary with time, that will affected by the genetic or epigenetic factors or change in the culture medium composition. So next is, you will get 2 marks question, what is mean by meristomoid. So what happens when the explant which is inoculated on the medium contains the hormone, the explant also contains a plant growth hormone that will induce the dedifferentiation phenomena. And what happens, there will be formation of meristem. So that will form the random cluster in the callus tissue, and that is named as that is known as a meristomoid. So you will get 2 marks question. You can define it as the meristem in a callus are scattered and random. The appearance of localized cluster of cambium-like cell, those are termed as a meristomoid or nodules or growing centers, and that can give rise to shoot, root or callus or embryo. Then next is habituation of callus tissue. So you will get question on this also, what is meant by habituated callus. So what happens, to form a callus or formation of callus tissue, there is need of plant growth, that is auxin and cytokinin, and this callus tissue is maintained through the serial subculture. That's mean you are going to transfer the callus tissue into another media, another freshly new media. So that is serial subculture. And what happen, as the time passes, this callus tissue can synthesize their own plant hormone, that is auxin and cytokinin, so that it will not require exogenous addition of plant growth hormone, that is habituation of callus tissue. So you will write this definition, that is it's the capacity to synthesize amount of auxin and cytokinin which probably accounts for their independence of exogenously supplied hormones. So this is the habituation of callus tissue. And this property of the callus tissue is known as habituation, and the callus tissue is known as a habituated callus tissue. It's like a cancer cell. The habituated callus tissue does not require the exogenous addition of plant growth hormone. They can synthesize their own plant growth hormone. That is habituation of callus tissue. Then chromosomal variation in callus tissue, what happens? So there is chromosomal variations are generated into the callus tissue. So that's why the direct organogenesis or direct embryogenesis is preferred. This variation can introduce the new species in the environment. So that's why the direct type of organogenesis and embryogenesis is preferred, and this chromosomal variation can be genetically or epigenetically happens in the callus tissue. So epigenetics means that is external environment around the callus tissue, and genetic basis is due to the endoreduplication. What happens due to endoreduplications, the ploidy level of plant cell will change. That will change from diploid to tetraploid or hexaploid. That is the basis of chromosomal variation. So that will generate the somaclonal variation into the plant. The last point is significance or application of callus tissue culture. So this callus tissue culture can be used for to study the callus tissue is used for morphogenetic and physiological studies. Then it can with the help of callus tissue, one can regenerate the whole plant through the manipulation of nutrients and hormonal constitution. Then it can be used as an explant for somatic embryogenesis. You can use the callus tissue for the genetic or karyotypic variability. so that one can regenerate the plant from variable cells, that is having the different plody level. Then for cell suspension culture, the callus tissue can be used for a single cell culture. Then the callus tissue is also used for secondary metabolite production, and it is used for biochemical assay. So these are the applications of callus tissues. Thank you.